Lesson 1: Virus Detection and Isolation. This content is part of the module MBS 7117 Molecular Microbiotechnology, and it's housed in the Department of Biomolecular Lab Technology and Microbiology, abbreviated as BLT, in the School of Biosecurity, Biotechnology, and Laboratory Sciences, abbreviated as SBLS, in the College of Veterinary Medicine, Animal Resources, and Biosecurity, abbreviated as COVAB, in Makerere University. I am your lecturer, Dr. Penina Nsamba. So let's go back to our lesson 1, Virus Detection and Isolation. We shall start with part 1, which is the rationale for virus diagnostics and an overview of the basic methods for virus diagnosis. Our contents for today will be number 1, rationale for virus diagnosis, and 2, an overview on the basic methods for virus diagnostics. Learning objectives 1 and 2. By the end of this section, you will be able to, number 1, justify virus detection and identification. Number 2, outline methods for virus detection. So let's look at the justification for virus detection and isolation. First of all, we want to get an accurate diagnosis of clinical disease for purposes of clinical management or for management of an outbreak situation. We want to confirm the presence of the virus, and this is for purposes of further decisions for medical care, contact tracing, quarantine, or if we want to give permits for trade to occur. Then our second justification for identification of viruses is to determine the disease severity and also prognosis. So we can quantify virus load, and then we are able to determine our course of action. For example, if you want to give antiretroviruses. Thirdly, we want to monitor disease progression, and in this case it's a virus disease, or we want to monitor the response to an intervention, or even monitor recovery from a virus disease. So in this case, our patients may be on antiretroviruses, and we want to see whether we want to take the treatment to a higher level or decrease the treatment if our patient is recovering. Or in case of very infectious viruses such as Ebola, we want to ascertain that our patient has recovered fully before they can be integrated into the communities. Fourthly, we do virus detection and identification for purposes of epidemiological surveillance. We are tracking the origin and spread of viruses, or in case of vaccination campaigns, we are tracking the success of this campaign moving towards eradication. Still on the justification of identification and detection of viruses, we are looking at infection control and biosecurity during the management of an outbreak. So if we are carrying out disinfection or decontamination, we want to ascertain how thorough the process is. Then we'll always want to screen our biological products for safety and make sure they contain no viruses. These are products like blood for transfusion, serum, antiserum, among others. We are looking at research and vaccine development. So if you're going to study virus properties, interaction of viruses with cells, response to antivirals, among others, we need to know what we are dealing with. Hence, we need to detect and identify a virus in question. Now let's look at an overview of various methods used for virus detection. In the picture here, we have rapid diagnostic tests where we collect a sample and we want, perhaps even at the point of source, to be able to know what virus we have. Or we can have blood samples at the point of care and using CRISPR actually find out exactly there what virus we are dealing with. So the method we are going to use to detect the viruses will depend on various outcomes or various issues at hand. This includes the infection present, the turnaround time, that is how soon we want the results, samples collected, the sensitivity or accuracy of the test, the resources we have, and the diagnostic laboratories available. Our methods for virus detection can be either 1. Direct, where we are targeting the virus particle, or indirect, we are targeting antibodies produced by the host as a result of the virus infection. So let's look at our first method used to detect viruses, and this is an electron microscope. Our picture there shows somebody seated at an electron microscope on the topmost row, while at the bottom we see an output from an electron microscope. So the electron microscope was first launched in 1932, and up to date it is used to study virus replication or the analysis of viruses entering the cells and how they assemble into the cells. The advantages of this method of detection is that it is rapid. You get the results immediately. It doesn't require organism-specific reagents for recognizing the virus particles, and this goes for primers, protein assays, which need specific reagents. Our second method is X-ray crystallography. So some virus can be easily purified directly from their hosts or from tissue culture, and what we get after this purification is what we see on the left, a structure of the viruses. And the principle of operation is that X-rays are directed at purified virus particles which have atoms arranged in repeating patterns. And these repeating patterns are recorded and analyzed to reconstruct the virus anatomic structure, just as we see in this diagram, which is got from the foot and mouth disease virus. So the advantages of X-ray crystallography is that we can get the three-dimensional structures of the viruses revealed, and the protein components can be reconstructed with high precision. It is the foundation for drug discovery. The disadvantages is that it cannot work for viruses with poor infectivity, such as the hepatitis E viruses. And secondly, the biosafety concerns when handling infectious viruses, which have to be grown to high titers. Our third method is antigen detection. So virus proteins, which are antigens, can be detected in samples from the host. The samples may be saliva, nasal swabs, body fluids, among others. So this picture shows a common method of antigen detection, which is an ELISA assay. And the antigens that we detect in the sample can now be confirmed using, for example, an ELISA, enzyme-linked immune absorbent assay, which we've just shown in the picture, IFA, immunofluorescent assay, immunochromatogenic assay, lateral flow assays, or other rapid diagnostic tests. The advantages of antigen detection would be the results we get are rapid and it's cheaper than the PCR. Disadvantages: it's best used in early stages of infection when there are high viral loads. So in the later stages of infection, when the viral loads decrease, it doesn't give you good results. And another disadvantage is that it's less sensitive when you compare it to other methods like PCR. So it may not detect low viral loads, whereas PCR may be able to detect these low viral loads. So let's look at our fourth method of virus detection, which is using the molecular tests. We may be familiar with the PCR machine, which amplifies nucleic acids in the virus. And now these nucleic acids can be detected using an agarose gel. And we can even take the nucleic acids for sequencing. So to confirm the virus nucleic acids, we can use the polymerase chain reaction, which is a PCR. And it's sometimes coupled with reverse transcriptase if we are looking at RNA viruses. We can use real-time PCR. We couple it with nucleotide sequencing to know the different bases that make up the genetic nature of the viruses. And this nucleotide sequencing can be the Sanger methods, next generation NGS, or even whole genome sequencing. We can use loop-mediated isothermal amplification, the LAMP, or we can use CRISPR-based diagnostics at point of care to detect the nucleic acids. Advantages of molecular techniques: there's a high turnover rate and you get your results just in hours. So it's very helpful where the only methods previously we had before were to grow these viruses in cell culture, which can be slow for some viruses. It's very sensitive. Very small amounts of nucleic acid material can be detected in the virus and in the samples that have been submitted, making it very sensitive. It can detect viruses very early before antibody production has occurred. It can be automated, allowing us to analyze large numbers of samples. And you can detect multiple viruses simultaneously if you use the multiplexing options. The disadvantages of molecular methods is costly. Setting up the infrastructure and the expertise takes up a lot of capital. And secondly, it cannot differentiate between replicating virions, which are causing active infection, and nucleic fragments which are present in the host that are not capable of replication. So in short, we're not going to be able to distinguish between active infection and infection where we don't have replicating viruses. Our fifth method is the culture of viruses. So viruses can be grown in laboratory conditions which is also in vitro, using living cells. For example, cell culture. We can isolate viruses in a monolayer or in suspension, or we can use embryonated egg cultures, fertilized eggs inoculated with viruses, or we can passage the viruses into living animals. Mice are common, but we also can use guinea pigs or rabbits. The advantages of culture viruses. One, is a gold standard. It proves that we've got virus replicating. Secondly, it permits direct study of viruses, which includes their phenotypic characteristics like drug resistance, or perhaps how they produce plaques in cell culture, or how they produce clinical symptoms in animals. Third advantage is that antibodies are produced, which can be studied. And fourthly, it's used for vaccine production, especially the embryonated eggs. The disadvantages: it's time consuming. If you have slow growing viruses such as the HIV virus, it's going to take you weeks to get a positive reaction. Secondly, it's going to involve large quantities of replicating viruses, making it necessary to use the biosecurity level or BSL level 3 or 4 facilities. This makes it very costly, and this in turn poses a high biosafety risk and also biosecurity risk. And the third disadvantage is that when we look at protection of human rights, it doesn't support the use of experimental animals. So now we're going to look at indirect methods of virus detection. We are not detecting the viruses, rather we are going to detect antibodies. So we are going to look at mostly serological tests. So the antibodies, which are IgM or IgG, produced as a result of the virus infection, can be detected in serum samples. And the picture here shows an antibody. It's a diagrammatic representation of an antibody, while below we see a rapid diagnostic test where serum has detected a positive reaction to the antibodies. So let's go back to antibodies, and we said they can be detected using the following assays: enzyme-linked immune absorbent assay or ELISA, immunoblotting such as the Western blot, and rapid diagnostic tests which confirm the presence of antibodies, just like what we've shown you on the left. The advantages of serological tests: we can determine previous exposure to viruses, even if the virus is not present. It can be used for viruses that cannot be cultured, or in cases where direct virus detection is impractical. The disadvantages: it cannot differentiate between active infection and past infection. And in some cases, it may not be possible to differentiate the vaccinated animals from the infected animals. Our third disadvantage is that there may be cross-reactivity, leading to many false positives. Our second method of indirect detection of viruses is by use of the neutralizing antibody tests, or virus neutralizing assays. So we are detecting virus-specific neutralizing antibodies in the serum. And these antibodies can block the virus from infecting cells. So if we have got a 96 well plate cultured with viruses, a 96 well plate cultured with cells, and we put on virus, and then we put on neutralizing serum, would want to say that there will be some places where you'd see cytopathic effect and therefore no cells, in contrast to some areas where there is no cytopathic effect. So our cells would be present, meaning that the antibodies in the serum have blocked the virus from infecting the cells. So just briefly, this method, which we are going to see later, would mean that when we have this 96 well plate with a monocell culture growing, and then we put on our serum that contains neutralizing antibodies towards this virus. When we eventually inoculate virus on this 96 well plates, we're going to get this picture. Some parts, the antibodies in the serum has prevented the virus from infecting the cells. So let's look at the advantages and disadvantages of this method. The advantages is that it's the gold standard. It measures immunity provided by vaccine or infection with a virus. The disadvantage, it's lengthy and time consuming, and it needs staff that have been well trained. So let's look at our self-reflection questions. Our first question would be, which of the following is a molecular diagnostic technique? Let's underline molecular diagnostic techniques. So we're not looking at antibodies. Neither are we looking at virus particles growing. So, which of the following is a molecular diagnostic technique? And yes, it's PCR. Our second question, which of these diagnostic methods is most suitable for detecting antibodies against HIV? And let's underline detecting antibodies against HIV. So you've got it right, it's ELISA. Third question, which of the direct diagnostic methods can detect a virus directly from a patient's sample without requiring virus growth? So we are not doing detection of antibodies. Rather, we are detecting viruses without cell growth, or virus particles without cell growth. And yes, you've got it. It is PCR. So let's move on to the next question. Question 2. Compare and contrast virus culture in cells and molecular diagnostic tests in terms of sensitivity, specificity, turnaround time, resources available, laboratory settings, and manpower. And just a reminder, we are looking at virus culture in cell lines and molecular diagnostic techniques. So ideally, this will be a take-home exercise. Now, in summary, what we've learned are the rationale and basics of virus detection and identification, which include justification, different methods, and factors influencing the choice of methods. The next steps, please refer to the lecture notes that we have given you on the study portal and the ETVTs and the chapters in the textbooks that we have recommended to you for further reading. The following has been our source of information. And thank you for your attendance.